Imagine trying to put out a fire in a specific room of this massive, sprawling mansion.
Okay.
But as you approach it, you realize the mansion is actively building barricades to block your path.
Right.
And it's pumping out this toxic gas that literally melts your fire hoses. And even crazier, it's actually brainwashing your own firefighters to turn around and defend the flames.
Yeah, which is an absolute nightmare scenario.
Right. But that nightmare, that is exactly what treating ovarian cancer is like today.
It really is.
So, welcome to this deep dive. We are taking a really hard look at a disease that's often known as the silent killer. I mean, ovarian cancer, it has a 5-year survival rate of just around 45%.
Yeah, and a recurrence rate that sits stubbornly over 70%. It's incredibly difficult to treat.
Exactly. But today, we are unpacking how we are on the absolute verge of treating this with a technological approach that, honestly, it feels like it was ripped straight out of a science fiction movie.
Oh, totally. I mean, it really is a complete reimagining of medicine. We are moving away from this sort of brute force approach, and we're looking at a highly targeted, almost surgical infiltration of the disease.
And to guide us through this, we have an incredibly dense, cutting-edge review paper. It was published in partnership with the Hormel Institute at the University of Minnesota.
Right. A really fantastic team over there.
Yeah. And our mission today for you, the listener, is to untangle this research. We're going to explore how 3 distinct technologies, so that's spatial omics mapping, biomimetic nanoplatforms, and artificial intelligence, how they're basically being fused together to infiltrate and destroy the localized defenses of ovarian tumors.
And, to really grasp why this triad of technology is so revolutionary, we first have to understand why current treatments are just, failing.
Right.
Because traditional immunotherapies, specifically immune checkpoint inhibitors, they're hailed as miracles for some cancers, right?
Like melanoma, I think.
Exactly. But in ovarian cancer, their objective response rate is dismal. It's less than 15%.
And that goes back to that nightmare mansion scenario I mentioned. We are flooding the patient's system with these really powerful drugs, but the drugs physically cannot reach the target.
Because the tumor builds a fortress.
Exactly. So let's start there. How exactly is this tumor protecting itself from our best medicines?
Well, the primary obstacle is not actually the cancer cells themselves, interestingly enough.
Wait, really?
Yeah. The real problem is the neighborhood they construct around themselves. So in oncology, this is called the tumor microenvironment, or the TME.
A TME.
All right.
Yeah.
And the defining feature of the ovarian cancer TME is its intense spatial heterogeneity.
Which basically means the tumor isn't just a, uniform lump of identical rogue cells.
Not at all. No, it is highly organized. It has specific, localized defensive zones. And the most insidious part is how the cancer recruits normal, healthy immune cells, pulls them into these zones, and completely corrupts them.
It essentially drafts an army of guards. Okay, let's look at the roster of these guards. The paper highlights 3 major culprits here. What are we dealing with?
First, we have what are known as Tregs, or regulatory T cells.
Okay.
Now, in a normal, healthy body, your Tregs are the peacekeepers. So after your immune system fights off a cold, the Tregs tell the killer T cells to calm down so they don't start attacking healthy tissue.
That makes sense.
But inside the tumor's microenvironment, these Tregs are hijacked. They express specific markers, you'll see them, called FOXP3 and CTLA4 in the literature.
Whoa, okay, alphabet soup alert right there. Let's break that down for the listener. What did those markers, FOXP3 and CTLA4, actually do to the immune system?
Think of those markers as like a VIP badge. It gives the Tregs the absolute authority to shut down the party.
Oh, I see.
Yeah, by expressing FOXP3 and CTLA4, these corrupted Tregs actively suppress the good tumor-fighting T cells. They act like bouncers standing outside the cancer cells, just, turning your own immune system away.
That is incredibly frustrating. And they aren't the only ones doing this, right? What's the second type of guard?
Those would be TAMs, or tumor-associated macrophages, specifically a subset called M2 type.
Macrophages. Okay, so normally macrophages are like the garbage collectors of the immune system, right? They eat dead cells and debris.
Exactly. But the tumor releases these specific chemical signals, like IL-10, that literally brainwash these macrophages. And once they're corrupted into M2 TAMs, they start expressing proteins like ARG1 and VEGF-A.
And what's the real-world consequence of those specific proteins?
They do two really devastating things. First, they promote further immune evasion, basically hiding the tumor.
Okay.
And then VEGF-A specifically triggers the construction of new blood vessels.
Wait, so the brainwashed garbage collectors are now literally building infrastructure to feed the tumor?
Yes, exactly. They're laying down supply lines. And then you have the third guard, which are MDSCs, or myeloid-derived suppressor cells.
And what do they do?
Their mechanism is just brutal. They release reactive oxygen species and nitric oxide directly into the immediate area.
So they're literally pumping toxic chemical gas into the environment to melt the weapons of any attacking immune cells.
That is exactly what they're doing.
Man. So we have bouncers turning the good guys away, construction workers building supply lines for the tumor, and chemical warfare rendering our drugs totally useless.
It's quite the fortress wall.
It really is. But the paper also talks about a vertical barrier, right? Like a moat around this fortress. And that moat is built out of hypoxia, or like a severe lack of oxygen.
Yeah, hypoxia is perhaps the ultimate defensive trigger. As a tumor grows rapidly, it outpaces its blood supply, which creates these regions right at its margins that are totally starved of oxygen.
Okay.
And this drop in oxygen activates specific oxygen-sensing regulators in the cells, primarily one called HIF-1 alpha.
HIF standing for hypoxia-inducible factor.
Yes. And when HIF-1 alpha senses that oxygen is low, it hits the panic button and constructs an immune barrier.
How does it do that?
It does this by upregulating something called PDL1 on the surface of the tumor cells.
Okay, I've heard of that.
Yeah, PDL1 is essentially a fake ID. It tells the attacking T cells, hey, I'm a normal part of the body, stand down.
Wow.
And even worse, another regulator, HIF-2 alpha, forces the tumor cells to broadcast a distress beacon. It's a chemokine called CXCL8.
And I'm guessing that beacon doesn't call for help from the good guys.
Definitely not, no. It acts as a magnet for more of those corrupted TAMs and toxic MDSCs. It pulls them right into that oxygen-starved moat, creating this vicious, self-reinforcing feedback loop.
So they're just piling in.
Yeah, and they don't just sit there in silence either. There is constant, incredibly complex intercellular chatter. For instance, the TAMs use a signaling axis called SPP1-CD44.
Let me guess. Another mechanism to neutralize our immune system.
You guessed it. The SPP1-CD44 signal directly forces any nearby active T cells to put up exhaustion markers.
Exhaustion markers?
Yeah, things like LG3 and TCD. It's essentially a localized radio frequency that literally puts the good guys to sleep. And there's also the CXCL12, CXCR4 axis, which organizes the physical layout of all these corrupted cells so they overlap perfectly to create maximum resistance.
Okay, so let me just do a quick recap for you listening to make sure we're all getting the full picture of this nightmare.
Go for it.
So ovarian cancer creates a highly localized heterogeneous fortress, right? The TME.
Yep.
When it runs out of oxygen, it uses that hypoxia via HIF-1 alpha as a trigger to throw up fake IDs. Then it uses a chemical radio network to recruit brainwashed macrophages, toxic gas pumpers, and bouncers to form this impenetrable wall around itself.
That is the ovarian cancer tumor microenvironment in a nutshell, yeah.
So think about this for a second. If you're a listener thinking about this from a medical perspective, how do you even begin to attack a fortress like that? I mean, you can't just throw a systemic drug at it, right? It'll get neutralized in the moat. And if you can't even see the individual walls and guards, you're fighting blind. Which brings us to the first massive breakthrough in this research. We are finally building the map.
Because you cannot defeat a fortress with a blurry big picture photograph from an airplane. You need high-definition, street-level GPS.
Exactly.
And in this context, that GPS is single-cell and spatial omics.
Okay, let's start with how we look at just one individual cell. Because traditionally, if I wanted to study a tumor biopsy, I'd take a piece of tissue, grind it up, and look at the genetic material, right?
Yes, bulk sequencing. But think about what that actually does. If the tumor is a fruit basket, bulk sequencing puts the entire basket into a blender and gives you a smoothie.
Okay, I like that analogy.
Yeah, so you might know there was some apple and some banana in there, but you have no idea how many or what state they were in.
Right.
The paper details a shift to cRNA-seq, that's single-cell RNA sequencing. This isolates every single cell and quantifies its specific messenger RNA.
Which is basically the active instruction manual the cell is reading at that exact moment.
Precisely. By reading the mRNA, we capture the dynamic state of that individual immune cell. We can see in real time whether a T cell is actively fighting, whether it's migrating, or whether it has been put to sleep by the tumor. Wow. But the researchers didn't stop at the RNA. They also wanted to look at the physical structure of the cell's DNA using SCATAC-seq.
Epigenetics.
Yes. And this technique uses an incredibly precise enzyme called TN5 transposase.
Okay, explain how that works.
Well, imagine a cell's DNA as a massive, tightly wound spool of millions of pages of instructions. Most of it is closed up, totally inaccessible. TN5 transposase acts like a highly specialized librarian that only searches for the sections of the DNA that are unspooled or open.
Because if a section is open, it means the cell is actively using those specific genes. It's like finding a bookmarked page in a massive encyclopedia.
That is exactly the concept.
Right.
By mapping these open chromatin regions, researchers found the actual epigenetic vulnerability.
What did they find?
They discovered that binding sites for things like AP1 and STAT3 are specifically cracked open and hyperactive in the corrupted CAMs of ovarian cancer patients.
So we don't just know that the cell is behaving badly, we know the exact line of code driving the bad behavior.
Right.
Okay, but I have to push back here for a second, though.
Sure.
I understand the power of looking at a single cell's instruction manual, but if we are isolating single cells, if we are physically pulling them out of the tumor to sequence them, we lose their location.
Yeah, we do.
We know who the enemies are, but we have no idea where they are in the 3D space of the fortress. We've lost the map.
And that is the critical flaw of single-cell sequencing on its own.
Yeah.
And it is the exact problem that spatial transcriptomics, or ST, was invented to solve.
ST?
Yeah, this is the real game changer.
Yeah.
ST platforms take all of that genetic chatter we just talked about, and they overlay it directly onto an intact physical slice of the tumor tissue.
So we aren't grinding anything up. We got the physical location coordinates and the genetic data simultaneously?
Yes, exactly. The review paper highlights several platforms making this possible. You have 10x Visium, which gives a resolution of about 10 to 100 micrometers.
Okay.
Then you have SlideSec, which uses these tiny nanobead arrays to map those specific signaling hotspots like that SPP1 radio frequency we mentioned earlier.
Wait, the sleep signal.
Yeah. And then you have StereoSec, which pushes into subcellular precision.
Wait, subcellular, meaning we can map out what is happening inside specific quadrants of a single cell on a physical map.
We can. It's truly wild. And when researchers applied this spatial mapping to ovarian tumors, the results were striking.
What does it look like?
They proved visually that those corrupted Tregs and M2 TAMs don't just float around randomly. They cluster perfectly at the boundary between the tumor and the surrounding stroma, and they directly overlap with the HIF1 alpha zones.
Oh, wow. So the guards are standing exactly at the edge of the hypoxic moat.
Precisely. And we can actually use software to trace the communication lines between them.
Like tapping their phones.
Exactly. We can map the network topology. Using bioinformatic tools like CellPhoneDB, NicheNet, and CellChat, researchers analyzed this spatial data and found that the TAMs serve as the central hub nodes for the immune suppression.
Okay, so if the tumor is a criminal syndicate, the TAMs are the local bosses handing out all the orders to the bouncers and the chemical gas pumps.
That's a perfect way to look at it.
So, okay, let's recap this second subtopic. We've gone from essentially blending the tumor into a smoothie to having a microscopic 3D GPS map.
Yes.
And this map shows us exactly where the moat is, exactly where the mob bosses are standing, and exactly what instructions they are reading using things like cRNA-seq and spatial transcriptomics.
We have the exact coordinates.
Which raises the next massive question for you listening. Now that you have these incredibly precise 3D coordinates, how do you deliver a weapon directly to the mob boss without destroying the healthy tissue nearby?
Because systemic chemotherapy won't work.
Right. We need a vehicle that can navigate the moat. Enter the Trojan horses.
Yeah, traditional delivery just doesn't work here. The TME fortress blocks it, or the drugs cause massive toxicity throughout the rest of the patient's body. So the paper solution is the development of biomimetic nanoplatforms.
Biomimetic meaning it mimics biology. We are building synthetic nanocarriers, but disguising them as natural biological components.
Yes, and the paper explores three fascinating ways to do this. The first is cell membrane-coated systems.
How does that work?
This is where scientists take a synthetic nanoparticle loaded with a drug, and they literally wrap it in the membrane of an actual T cell or red blood cell from the body.
Oh, wow. I see it now. It literally is a Trojan horse. It's wearing the enemy's uniform.
Yeah.
So when it approaches the tumor, the bouncers look at it and say, oh, you're just a red blood cell. Go right ahead.
Exactly. It completely evades the body's immune clearance and eerily penetrates the fortress. The paper refers to this as achieving homologous targeting.
Homologous targeting. Got it.
The second type utilizes exosomes. Now, exosomes are naturally occurring nanovesicles. They're these tiny packages that cells normally use to send proteins and RNA to one another.
Oh, so researchers can hijack these natural biological FedEx trucks, empty them out, and pack them with therapeutic drugs instead.
That is brilliant, using the body's own mailing system against the tumor.
It really is. What about the third type?
Those are artificial hybrid systems. These are usually liposomes, tiny fat bubbles that are heavily modified with specific targeting peptides.
Okay, give me an example.
For example, the paper discusses a peptide called CRV. When you attach CRV to a liposome, it acts like a highly specific homing beacon that only binds to those corrupted M2 TAMs.
The mob bosses.
Right. And the payload it delivers doesn't just kill the TAM. It converts the corrupted M2 type back into a healthy, tumor-fighting M1 type.
It brainwashes the guards to turn around and start fighting for us. That is incredible.
It's reprogramming at a cellular level.
But, here is a detail from the paper that I found absolutely mind-blowing. These nanobots aren't just dumb capsules floating around bumping into things. They are, quote, multi-responsive, meaning they are programmed to detonate only when they encounter specific environments.
And this is the true genius of the engineering. They take the tumor's own localized defense mechanisms and use them as the very triggers to open the Trojan horse.
So cool.
The paper outlines 3 main trigger mechanisms. Let's look at hypoxia-responsive triggers first. Researchers use specific chemical structures like azo-aryl groups or 2-nitroimidazole.
Okay.
These structures hold the nanoparticle tightly together, but they are incredibly unstable in low oxygen environments.
So the moment they float into that oxygen-starved moat driven by HIF-1 alpha, the chemical bonds physically snap.
Exactly. They undergo cleavage, breaking apart and releasing the drug only where the hypoxia is highest.
That's amazing. What was the next one?
A second trigger mechanism is enzyme responsive. See, the tumor stroma is constantly secreting specific enzymes to remodel the tissue around it. Things like matrix metalloproteinases or MMPs.
Okay, the construction workers.
Right, so researchers engineer the nanocarrier using a specific peptide sequence called GPLGIAGQ.
Well, and guess, that sequence is the exact thing the MMP enzyme likes to chew up.
Yes, the nanocarrier remains completely solid while traveling through healthy tissue. But the second it encounters the tumor's specific MMP enzymes, the sequence gets severed and the payload drops.
Wow.
And the third trigger is pH responsive.
Because tumors have a warped, highly acidic metabolism compared to healthy tissue, right?
Right. The core of a tumor is mildly acidic due to lactic acid buildup. By building the nanoparticles out of specific polymers like PLGA or PEG-BPDMA, the particle acts like a chemical sponge.
Okay.
It ignores neutral pH, but when it hits the acid of the tumor, the polymer physically swells up until it bursts open.
So clever. And what exactly are we packing inside these responsive Trojan horses?
Well, because the tumor has multiple layers of defense, the paper highlights co-delivery strategies. You need a coordinated attack.
Like a combo move.
Yeah, exactly. One great example is delivering an anti-PD-L1 agent to tear up those fake IDs and wake up the exhausted T cells right alongside a drug called lactate oxidase.
And what does that do?
Lactate oxidase specifically eats up the acidic waste the tumor produces. So you are destroying the chemical shield and arming the immune system at the exact same physical coordinate at the exact same time.
Okay, let me recap the strategy so far, because it's a lot.
It is a lot, yeah.
So we use biomimetic nanocarriers to sneak past defenses. We engineer them with specific peptides, like CRV, to target corrupted cells. And we program the drug to only activate when it senses the specific acid, enzymes, or lack of oxygen unique to that tiny tumor neighborhood.
The precision really is staggering.
It is. But as I was reading this, a massive problem occurred to me. Biology isn't static, right? A tumor mutates. It adapts. So, for the listener, think about this. What happens when the tumor adapts and changes its defenses mid-treatment? If your multi-million-dollar Trojan horse is programmed to burst open at a specific pH and the tumor alters its metabolism so it's no longer acidic, your nanobot is suddenly completely useless.
And that realization is what brings us to the final and perhaps most crucial piece of the puzzle. If we rely on static delivery, we will always eventually lose to adaptation.
Right.
We have to move from static delivery to dynamic response. Our delivery system needs a brain.
Which brings us to the final technology, artificial intelligence. How do we put an AI commander in charge of these nanobots to keep up with a mutating tumor?
We shift to an active, closed-loop system.
Closed loop.
Yes, the paper describes an identification delivery feedback optimization loop. The AI models act as the operational commanders. And the review highlights several specific frameworks currently in development.
Okay, let's hear them.
One is called Viz-AI. It uses convolutional neural networks to read the spatial omics maps we talked about earlier.
So it's looking at the pictures.
Right. It actually learns to visually recognize the heat maps of those corrupted Treg clusters, and it predicts the absolute most effective delivery zones for the nanomedicine.
So the AI is analyzing the battlefield map and plotting the exact landing zones for the Trojan horses to do the most damage.
Yes, and it gets deeper. There is a framework called drug spatial net.
Drug spatial net, what does that do?
This uses multitask learning. You feed all that dense omics data into the AI, and it spits out a highly specific recommendation. It tells you what kind of biomimetic platform to use, which combination of drugs to put inside it, and exactly where to send it in 3D space.
But wait, how does an AI connect a massive spreadsheet of genetic sequencing data to a physical picture of a tumor slice?
Through incredibly complex math, specifically algorithms like the Tangram algorithm.
Okay.
Tangram takes the single-cell RNA sequencing data, the instruction manuals, and mathematically aligns it pixel by pixel with the spatial images of the tumor to create a unified predictive model.
That covers the planning stage, though. But you mentioned a closed-loop system earlier. A true loop implies real-time feedback, right?
Right. And this is where we cross firmly into sci-fi territory. The paper details intelligent feedback regulation built into the nanoplatforms themselves.
How does that even work?
Well, imagine a CRISPR switch-controlled nanosystem. Unlike the triggers we discussed earlier that just burst open once and are done, this nanoparticle is dynamic.
Okay.
It is loaded with a drug, say, a TGF-beta antagonist, but it only releases a dose when it senses local levels of IFN gamma fluctuating in the environment.
Wait, hold on. IFN gamma is a marker of immune system activity. So you are saying the medicine itself is actively reading the patient's immune response in real time?
It is actively listening to the environment, yeah. It senses the immune system waking up and starting to fight. It releases just enough drug to support the attack.
Oh my God.
And when the immune response peaks, the nanoparticle actually pauses its release to prevent overactivating the immune system, which causes severe toxicity. When the immune response dips again, the nanoparticle senses the drop and releases more drug. It is a completely self-regulated, rhythmic release loop occurring at the microscopic level inside the tumor.
That is genuinely mind-blowing. A robotic swarm dosing itself rhythmically based on your body's minute-by-minute biological feedback.
It's the ultimate precision medicine.
But let's bring this back down to earth for a moment. What is the actual path to getting this into a human clinic? Because standardizing all this multimodal data, the RNA sequencing, the spatial maps, the complex AI algorithms across different hospital systems sounds like an absolute logistical nightmare.
It is the single biggest translational hurdle. I mean, a predictive AI is only as good as its training data.
Obviously.
So the 10x Visium spatial data collected from a hospital in New York has to be perfectly, seamlessly compatible with the AI models running in London or Tokyo.
Right, otherwise it breaks.
Exactly. To solve this bottleneck, the paper highlights a massive initiative, the proposed OVAMAP Consortium. The entire goal is to share cross-platform spatial transcriptomics data globally to create a national ovarian cancer spatial immune database.
Because without that massive standardized data set, the AI has nothing to learn from, and the robotic swarm never gets off the ground.
Precisely. The data infrastructure has to be built before the clinical trials can truly utilize the AI.
Okay, let's recap this AI section. So AI models like drug spatial net process all this omics data to predict optimal drug delivery coordinates.
Yep.
Then next-gen nanoplatforms act as closed-loop systems, actually reading biological feedback like IFN gamma to adjust drug release in real time. And massive cross-hospital databases like OVAMAP are required to standardize these AI predictions for real clinical use.
Perfect summary.
So think about this, listening to this. We are looking at a future where your cancer treatment isn't a static pill you swallow every morning. It is a spatially intelligent, self-regulating swarm governed by an AI that constantly adapts to the exact geography of your specific biology. What stood out to you the most about this closed-loop system? Take a second to really think about that paradigm shift.
It fundamentally changes how we view systemic medicine, doesn't it? For decades, the philosophy has been to flood the entire body with toxic chemicals, hoping to kill the cancer just slightly faster than we kill the patient.
It's brutal.
It is, but this research proves that we are moving towards spatially intelligent medicine. We are treating the microenvironment, not the macroorganism.
So just to summarize everything we've gone over today, we started out looking at the impenetrable hypoxic fortress of the ovarian cancer TME, a nightmare scenario of brainwashed immune cells and toxic gas. But we've mapped it out pixel by pixel using spatial omics. We've designed multi-responsive Trojan horses to navigate the moat and breach the walls. And finally, we put an AI commander in charge to read real-time feedback and direct the attack.
And as we wrap up, I want to leave you with a thought to mull over, expanding on this entire concept.
Let's hear it.
If we are currently perfecting the ability to use AI and biomimetic nanotech to infiltrate, map, and dynamically rewrite the corrupted microenvironment of an ovarian tumor, could this exact same closed-loop system eventually be used to re-engineer other complex, failing microenvironments in the human body?
Oh, wow. Because a hostile microenvironment doesn't just have to be cancer.
Exactly. Could we send these dynamically responsive Trojan horses into the brain to repair the degenerating neural tissues in Alzheimer's disease?
That would change everything.
Or could we program them to perfectly sense and reprogram sites of severe autoimmune inflammation without suppressing the patient's entire immune system?
Wow.
The fierce, complicated fight against ovarian cancer might just be writing the underlying code for the future of all human healing.
That is an incredible thought. The techniques we are using to dismantle a fortress might be the exact same tools we use to rebuild a crumbling city. Thank you for joining us on this deep dive. Keep questioning the world around you, keep looking for the maps hidden in the chaos, and we will see you next time.